This tutorial will provide a general overview of Tinkercell. Tinkercell comes with a tree of biological parts that you see on this left-hand side. This tree is generated from an XML file, and so you may edit the XML file to change the format of this tree. To insert a part from this tree, you simply click on that part and then click on the screen, and then escape. When you select an item on the screen, you will see all the tools that are associated with that item. For example, this particular tool will allow you to scale or rotate the item. The history window will allow you to undo any of the things that you have done. These other tools, namely the text attributes and the numerical attributes, will show the text and numerical attributes that were listed in the parts tree XML file. All the items that you see on this tree have their own numerical attributes and text attributes that come from the XML file that characterize each of those parts. Now let's create a simple network and simulate that network. So I'm going to insert 3 proteins: protein A, protein B, and protein C. As you can see, each of these proteins have their own numerical attributes and text attributes, such as sequences, molecular weight, concentrations. Now I'm going to insert a reaction from A to B and then B to C. So I will select a reaction from this tree of reactions. This tree of reactions is also loaded from an XML file, similar to the tree of parts. So we'll select a simple reaction, A to B and then B to C. Now when I select this reaction, I see in the stoichiometry. The stoichiometry shows me what is happening in this reaction. So this particular reaction consumes one A and produces one B, and that reaction occurs at the rate of J0.K0 times A. J0.K0 is an attribute or parameter associated with this reaction, which you can see here. When you click on the numerical attributes, you see J0. J0 is the name of this reaction. Similarly, we have J1.K0 and rate for this reaction. You may select both reactions and see the complete stoichiometry matrix and all the rates, and edit any of those values. You may also see the K0 values here and change them if you desire. You can also set the concentrations of the proteins here. So I'm going to set the concentrations to 0 for BC, and then I'm going to simulate using the function here. This box contains several functions that are either C or Python functions. If you have programming experience, add your own functions to Tinkercell. So the deterministic simulation, press enter. It opens this new window that will ask you for the time, the step size, and whether you want to simulate the complete model or just the selected items in the model. So we will simulate the complete model and see this result. If you actually want the data values that are plotted here, Tinkercell informs you that data values have been recorded in this out.tab in your Tinkercell directory. And now the stochastic simulation. Similarly, there is our stochastic simulation. So those are 2 of the functions from this list, 2 of the more useful functions. Now let's construct a simple genetic network, since Tinkercell has several genetic components, and simulate the genetic network. The simplest way to build a genetic network is to use this composite part, which contains a promoter, a ribosome binding site, a gene, and a transcriptional terminator. Now I'm going to insert a transcription factor, or just a protein, that's going to control this promoter, regulate this promoter. Again, I'm going to insert a molecule that is produced from this gene. So the reaction going from protein to the promoter will be a transcriptional regulation, which is this particular reaction. From B to the promoter, and then the transcription of particular reaction, transcription from this gene to C. And as you can see, when you look at the stoichiometry for that particular reaction, it includes transcription translation. It includes the mRNA step. You may delete these intermediate reactions if you choose. They are just placed by default. And I'm going to insert the degradation reactions for and C. And then simulate this network. Deterministic simulation. Let's change the values slightly so that the deterministic simulation looks better. I'm going to change J1's K0 value to something higher, such as 20. And there is the simulation, and there is the stochastic simulation.